So, Saman, you are live.
Hello, my name is Saman Honarnejad and I'm Director of Drug Discovery at Pivot Park Screening Center, an independent contract research organization based in Os in the Netherlands that specializes in early drug discovery services, including cellular and biochemical assay development, ultra-high throughput screening, and hit-to-lead biological profiling. Ultra-high throughput screening, or UHDS, is at the core of our business. Even though PPSC was established in 2012, we have over 2 decades of experience in screening at our site in Os, within companies such as Organon, Schering-Plough, and MSD. So as a relatively young company, we actually have a long-standing track record in developing tailored HDS assay cascades and generating high-quality HDS data to identify starting points for drug discovery. Here is our UHDS platform that consists of 3 pods interconnected through a conveyor belt with a traffic couple. We have the capacity to have up to 2.5 million compounds online. In the 12-sided plate preparation pod, you see the compound stores and 3 echoes for acoustic dispensing. And we routinely generate 100 to 200,000 data points per day. Of course, the system can handle multiple smaller focus screens in parallel as well. All the instruments around the robotic arms are on carts, and depending on the type of a given screen and its requirements, one can plug in appropriate instrumentation into the platform. The other advantage of the platform is that one can keep it up to date. In fact, we have been doing so over the course of the last 6 years. In 2014, we have integrated the flipper for measuring fast kinetics, such as membrane potential and calcium kinetics in 1536 well-play format. Later on, we introduced state-of-the-art dispensers, plate washers, and readers into our system. And most recently, we have integrated the RapidFlex for label-free UHDS using MALDI-TOF mass spec readout. With that quick introduction about PPSC, I'd like to start this talk by building a case on a huge problem in global healthcare, which is the fact that despite all the advances made in discovery and development of new therapeutics over the last decades, we are still scratching the surface in terms of targeting disease-associated genes. As you see in this Venn diagram, there is so much more space in the genome to target with drugs. Of course, part of the problem is the druggability of different classes of targets. Nevertheless, druggability is an evolving term. For example, back in the 90s, kinases were not considered druggable. But now we have many kinase inhibitors that work very well in clinic. But the other side of the problem is actually the innovation gap. It is common knowledge that innovation mainly starts with basic research in academic institutions. And many therapeutically relevant disease targets are identified in academia, plenty of which are reasonably well validated. But many of these targets remain largely unexploited. An underlying reason for this is that, on the one hand, many academics and small and medium enterprises don't have access to the right infrastructure, as well as high-quality compound libraries and expertise to find molecules that modulate these targets. And on the other hand, large pharmaceutical companies and VCs are mainly attracted to later stage de-risk projects. And this is the whole idea of the European Lead Factory, to fill this gap. This pan-European drug discovery consortium fills this gap by capitalizing on 2 pillars: the joint European Compound Collection, or JECL, and the European Screening Center, or ESC, that consists of multiple partners providing access to UHDS infrastructure, compound logistics, triage capabilities, medicinal chemistry, protein production, and structural biology. What is unique about the Jekyll is that it's a large collection of high-quality compounds, partly contributed by FBA companies as in-kind contribution, and partly synthesized by chemistry contract research organizations and academic chemistry labs in Europe to further expand the biologically relevant chemical space. And as an academic researcher or an SME in Europe with a novel target or assay, you could submit a target screening proposal to the consortium and get access to this unique drug discovery platform at no cost. This was made possible by funding from IMI and the European Union. On the right-hand side, you can see the chemistry partners that advance the public compound collection within the Jekyll, and on the lower left-hand side, the FPO partners that contributed part of their in-house compound library, together making a unique and large selection of diverse compound screening collection. On the top left side, you can see the partners within the European Screening Center, each with complementary assets. In this talk, I'm mainly focusing on activities at Pivot Park Screening Center within the ELF consortium that has to do with assay development, ultra-high throughput screening, data analysis, and part of the triage for public target programs. One of the major determinants of successful compound screening is the quality of the compound library itself. You can see an analysis on the FPS subset of the library, which shows the key physiochemical parameters of the Jekyll, such as molecular weight and A log P, that are typical of a lead-like library, centered around a molecular weight of 350 and A log P of 2 to 3. The fraction of sp3 hybridized carbon atoms also indicates a broad spread of three-dimensionality. As I mentioned before, the Jekyll is also diverse. Here you see a matrix of nearest neighbor histograms that shows diversity within the individual 7 subsets along the leading diagonal, and pair by similarities between subsets are shown in off-diagonal cells. Inter-subset diversity is consistently lower than inter-subset diversity. The inter-subset histograms are all very different from the intra-subset histograms. In other words, it is apparent that the diversity within each company subset is quite similar, apart from the single case where the distribution of the maximum Tanimoto similarity is right rather than left skewed. Moreover, the same study reference at the bottom of the slide used Bayesian modeling and data from Campbell to show that compounds in this library are predicted to hit a broad spectrum of biological targets. Collectively, these analysis shows the library is of high quality with respect to drug likeliness, diversity, and biological relevance. Over the course of the first phase of ELF, PPSC worked on 7 different disease areas and 8 different target classes that were all crowdsourced from academia and SMEs across Europe. You see on the left-hand side that the main disease areas were oncology, CNS and neurology, and infectious diseases. We also focused on diseases that are mainly not pursued by pharmaceutical companies, such as neglected tropical diseases. And also on the right-hand side, different classes of targets that were mainly enzymes, protein-protein interactions, and receptors, followed by ion channels, GPCRs, protein-DNA interactions, protein-RNA interactions, and chaperones. Thanks to these programs, you can imagine how much additional unique knowledge we've gained over the years by working on so many targets and assays within different therapeutic areas. As you can see on the left side, these target classes were mainly based on biochemical assays, but also cellular ones. Also on the right side, distribution of different readouts used are shown. This really shows PPSC has the capacity to support many conventional readouts. And I mentioned to you previously that since 2019, we also have the capability to screen label-free multi-TOF mass spec assays in UHDS format. From 2013 to 2017, we carried out 72 full-depth screens within the European Lead Factory Consortium. Perhaps it's worth emphasizing that in 2017, in combination with non-ELF commercial screens, we hit the record of 20 million data points. That is nothing short of HDS output from a typical and well-established pharmaceutical company. And for a small company like us, this was really quite an achievement. Of course, to be able to do so many screens using various readouts and assays, one requires maximum throughput. As you can see here, on average, we generate 100 to 200,000 data points per day in 1536 well plate format. And even on a few occasions, depending on the complexity of the assay and the length of incubation times, we reached close to 500,000 data points per day. Here, I really want to emphasize that such a throughput is absolutely crucial for a centralized screening facility like us, mainly due to rapid turnover of screening projects with various complexities. And we are constantly trying to optimize our screening operations to generate high-quality data from large libraries, both on the technical but also biological side. Every screen typically starts with a pre-screen phase to make sure the performance of the screen meets our acceptance criteria. This was actually 50 plates in our case. As you see, most campaigns were completed within 4 to 5 days, and typically the 5th day was to retest the failed plates. Other campaigns that took longer were rare and only cases where the assay was quite unstable and not robust enough in 1536 well plate format, and thus carried out in 384 well plates. Actually, what we learned along the way was that if an assay is not robust in 384 well plates, scaling it up to 1536 well plates doesn't help either. Nevertheless, this is a clear data set that shows UHDS using robust assays in 1536 well plate format is the best way to explore a large biochemically relevant space rapidly, efficiently, and at low cost in terms of screens per day. Of course, there are other alternatives like DNA encoded libraries, which requires further chemistry and synthesis. Having worked on these assays, target classes, and readout technologies, an obvious question is whether we see any relationship between the primary active rates and targets, or readouts, and the short answer is no. As you can see from the percent CVs on top of each box plot, there is quite some variation in percent active rates based on Z-score within each target class or readout technologies used. Of course, this is not surprising because within each target class, there are many other subclasses with different target protein structures, different catalytic activities, different subunits, etc. Nevertheless, what you learn from this is that primary active rate is a poor predictor of true hits, especially for challenging protein-protein interactions and protein interactions with nucleic acids. Also, the general assumptions with respect to superiority of certain readout technologies over others does not seem to be the case here. We also tried to explore whether we could find any generalizable trends in the numerical parameters extracted from the screens of 72 different programs that number of screening days, active rates, confirmed active rates, number of reordered compounds, number of compounds that were registered as preliminary hit list, number of compounds that were registered as qualified hit list, or number of compounds in LCMS that were qualified. And so by carrying out the principal component analysis, we found that only trivial parameters such as plate format or screen days are the main origins of variations across the programs. Therefore, the important question here is how to reliably and systematically deal with assay compound interferences and false positives when we have so many targets and assay readouts. So we asked ourselves if we can come up with a cost-effective, fast, and reliable framework to probe how every single assay behaves before going to screening phase. So together with the University of Dundee and Bioascent, we put together the Robustness Set compound collection that consists of different classes of assay interfering compounds, such as aggregators, redox, fluorescent compounds, luciferase inhibitors, reactive compounds, chelators, and colored compounds. And we also combined these compounds with a set of drug-like molecules from which we did not expect any obvious assay interfering properties, together with many DMSO controls. The objective to do so was to perturb an HTS-ready assay with these molecules to identify assay liabilities before doing the screen itself. What results from testing robustness set compounds is that you get an indication of false positives, you can improve your assay conditions, you can design the triage cascades that you may require down the road, and you can get some information about the reproducibility and plate effects. Also, using the clean compounds, one can get an indication of the hit rate. We also generated a robust data analysis pipeline to quickly evaluate the results from these perturbations. Here you see an example of a fluorescence polarization assay tested with two reducing agents, DTT and L-cysteine. On the x-axis are the different classes of molecules, as I explained in the previous slides, and on the y-axis is the percent of actives per each class. As you can see, with DTT in buffer, over 40% of redox compounds are active, and switching to a weaker reducing agent brings it down to below 20%. Of course, this doesn't help you with liabilities towards fluorescence or colored compounds, but it helps you decide which follow-up assays to use down the road for counter screens. Another example of this is a luminescence assay for an epigenetic target, tested with and without detergent. As you can see in blue bars, including 2% of tergitol as the detergent in the assay buffer reduces sensitivities to different classes of interferences. But sometimes it's just better to switch to a completely different assay and readout type to have a high quality primary assay. And in this case, an HTRF assay doesn't show any liabilities beside a few of compounds that were in the colored subtypes. I should emphasize again that this doesn't mean that one assay is better than the other, But the take-home message is rather the fact that quality of assays is context-dependent, and it is a function of target, compound, assay buffer conditions, and additives, and the molecular interactions between these molecules. Therefore, we have put quite some effort into developing tailored hit triage cascades to identify high-quality starting points with drug-like mode of action. On average, we employed more follow-up assays than primary assays, some of which originated from the public program owners and some developed from scratch based on liabilities found during assay development phase, making more than over 200 assays. In summary, I showed you how the European Lead Factory boosts drug discovery across Europe by crowdsourcing drug targets, bridging the innovation, expanding the chemical space, and creating an early drug discovery knowledge network across Europe. I also provided you with some evidence for context dependency of targets coupled with different readouts, and showed you that every target and assay is unique and should be treated as such. And last but not least, the importance of triage cascades and identifying assay liabilities as early as possible. There are many achievements from the first phase of ELF. The qualified hit lists provided to the program owners resulted in drug candidates, startups, preclinical development programs, publications, many patents, transfer of knowledge across partners and also program owners, and many business opportunities and collaborations. Moreover, the achievements made in the first phase of ELF resulted in continuation of the ELF concept through the second round of funding by IMI and EU. Since last year, within the European Screening Center Unique Library for Attractive Biology, or ESCOLAB, to carry out an additional 50 screens. There are a few changes compared to the first phase. On the FPS side, Servier, Grunenthal, and Medicines for Malaria Ventures joined, and chemistry CROs are mainly involved when the QHL is released and subsets of hits are resynthesized. And now the consortium actually accepts high-content screening proposals to screen subset of 50,000 compounds. So if you have a novel target or assay, feel free to take advantage of this unique opportunity by submitting a target proposal to the consortium. And with that, I'd like to thank my colleagues at Pivot Park Screening Center and also our partners within the European Screening Center and the whole consortium within European Lead Factory, as well as all of the public program owners. And thank you for your attention. I'm happy to take any questions.
Hello, Saman.